Hello and welcome to the Ori Spotlight Podcast. This is your host, Jason Foster. Got some special guests with me today. Many of you will know Farlan Veraitch, our founder and chief scientific officer. And we're lucky to have Hong-Ming Hu, the CEO of ImmuXell's, with us as well. So welcome, Hong-Ming and Farlan.
Hello, my name is Hong-Ming Hu. So I'm the founder and the CSO of ImmuXell's in Shanghai. Very nice to talk to you here.
And Farlan, welcome back. You're a multi-time visitor to the Spotlight.
Thanks, Jason. Excited to be on this particular podcast, particularly with Hong-Ming. Really excited about today's conversations.
Me too. I'm excited to have both of you. We recently, as some folks will know, announced our collaboration publicly, which has been going for about 5 or 6 months now. And the very, very exciting milestone that together with Hong-Ming and his team at ImmuXell's, we have treated the very first patient with a product that was manufactured in GMP on the IRO system. So very exciting milestone for us. I personally have been working towards this for the 7 years that I've been with Ori, and Farlan, it's been 10-ish for you to working towards getting the first patient treated. So a very exciting milestone that we're going to talk much more about today. But if we can, I'd like to start at the beginning and Hong-Ming, ask you to just introduce yourself a bit, if you don't mind. Give us a bit about your background. I know you were in the U.S. for 30 years, I think, before doing cancer research and T-cell therapy before moving back to Shanghai and working there. But could you give the audience just a little bit of your background and tell us about your career journey?
Jason, thank you. So as Jason already alluded, I've been in this field for almost, I think, 30 years. So 94, I went to a place where I started TCR therapy with one of the earlier pioneers in immunotherapy. So from there, we start to learn how to do adoptive cell therapy and also to find out how T-cell recognize the tumor. And then we're able to learn a lot from that earlier study and able to using that technology after I come back to China and get to patient treated. And now we think about a new challenge and then take that challenge with Ori and to scale up and then to reduce the cost of production. And really happy to able to do that with them. And then quickly we get first patient treated just 2 weeks ago.
I think you had mentioned that in the follow-ups, patient is responding as you would expect them to at this stage. And just for clarity, it's a pancreatic cancer patient. Is that correct?
We have been doing this with our own manual process and already and see some exciting earlier result. And we realize, the manual production is going to be a major bottleneck. So we thinking scale up and reduce cost is the way to go. And now we show initial first patient, we able to do that. And we are very happy to continue and doing this kind of automation and to see the safety and then efficacy of the cell we infuse patient with.
Yes, exactly. We'll come much more into that later. I definitely want to cover that. But let's just back up one step, if you don't mind, and tell us a little bit about ImmuXell's focus. So we're focused in solid tumors. And we know that, the audience will know certainly that solid tumors have been proven to be much harder to treat than liquid tumors, at least historically. And you've got a unique method to target the KRAS, the G12V mutation. So just tell us a little bit about how you ended up with that target and how kind of the unique elements of the ImmuXell's approach, if you don't mind.
Let me start talking about TIL therapy. So TIL, we been doing that for a while, and we don't know what the major T cell response targeting to. But one of particular patient early on we know is targeting KRAS mutation, and then we realized that might be a good target. And then KRAS is known being a driver oncogene, right? So we realized this can be a good target because it's more difficult to escape compared to a lot of other tumor-associated antigens. So the KRAS mutation is more frequent in pancreatic cancer. So that made me thinking we can using adoptive cell therapy for most difficult to treat cancer. And when we clone the one of particular T cell receptor in context of A11, we thinking maybe find more patients in China. So 21st, I come back to China, set up a lab, and then and to clone more T cell receptor from Chinese patient, and we was very lucky from TIL. We identified first T cell receptor, and then treat the first patient. As you probably know, Chinese have a IAT system which allow us to quickly doing few patient trial with a new CAR-T or TCR therapy. So we did that trial, IIT trial, 3 months after our first discovery. And then we see a great, very exciting response. Patient have a CR for 8 months for first patient. That laid the foundation for us to continue pursue this target. And then right now we clone many more T cell receptor against the mutation, including G12V, G12C. So we think we are in a unique position to do the therapy not only in more patient in China, also the speed we're able to testing new T cell receptor, new process for manufacturing, and all other target.
Thanks, Hong-Ming. So interesting kind of history of you moving back to China and identifying the opportunity to really accelerate development using the Chinese IIT system. So let's just delve a little bit into that a bit more, because I think it also connects to how we've been able to move so quickly together as a partnership. So you had begun testing your unique approach via the IIT system over the last year or more. And am I right that you had treated something like 12 or 13 patients before
Yes.
sort of we started working together? Okay. And then you're sort of iterating as you go with your unique approach. So tell us a little bit more about that. So you've got the unique target in the KRAS, G12V mutation, but you also have a unique vector, if I understand it correctly, that you're using. Is that correct?
Yeah, so that's very good point. So we always thinking about how we reduce the cost. And two major costs in cell therapy for CAR-T or TCR-Ts. One is the material cost, mainly the viral vector, right? So it's a major cost, and then another one is labor. So we think, using as a biologist, so I want to using our knowledge and tool to reduce the viral production costs. So we actually focus on retrovirus. So retrovirus a little different from the lengthy most people use. We're able to do a stable cell line to produce the virus continuously, and then that already easy for scale up and reduce cost later on. And then another innovation we think we are doing is try to make that system can target into T cell by engineer antibody specifically recognize and activate T cell, mainly CD3 antibody. So after probably a year and a half optimization, we able to modify our retroviral vector to be able to not only activate T cell, and at the same time transduce activate T cell. Uh, we call the one-step transduction because for retrovirus, typically don't infect non-active cells. So got two abilities of only infect T cell because we're using T cell antibody and not allow one-step activation and transduction. And in the end, it also leads to high efficiency because only activate cell can be transduced, only activate T cell can proliferate to in the end. So we typically get a really efficient transduction using our system, and then we're able to couple with Ori bioreactor to one-step transduction. That's really, I think, a beautiful marriage between the viral platform and then automated and then really consistent to driving costs of cell therapy in two front. One is viral factor, and another is labor.
Absolutely. Farlan, maybe I can ask you. I mean, we've worked with a couple partners that were using retrovirus, but not many. Most are using Lenti, as Hong-Ming said. Um, can you speak to kind of how the collaboration has worked over the last kind of year or so? I think Hong-Ming and I probably spoke early last year, early 2025, and then you guys began speaking on the kind of CSO to CSO level. You went out to Shanghai a couple times. Just talk us through the process of how we've kind of built the partnership over the last kind of year or so.
So, as everybody knows, Ori has a laser focus on driving down the cost of goods and enabling scale up, be that either centralized or decentralized. My first meeting with Hong-Ming was incredible. Every single piece of data that he showed me had a little box in the top right hand corner of the PowerPoint showing the cost of goods associated with that run. And I thought that was radical from a discovery team. I actually found it quite inspirational, my first meeting with Hong-Ming. He was innovating around the core biology of the transduction and activation step in order to drive down the cost of goods and the cost per run. He was making the manufacturing process more simple, more elegant, fewer steps. I think one thing which we haven't mentioned was that even in the virus manufacturing, Hong-Ming was thinking very carefully about how much was the cost of the virus. So he was innovating as well with the manufacturing process for the virus, making sure that that was going to be cheap. So when I came into the picture and we started to marry the biological and manufacturing innovation that Hong-Ming was driving, and we started to marry that with IRO's ability to de-skill, reduce the footprint, multiplex so that you can manufacture a lot of doses in a very small amount of space. Our ability to, really push manufacturing into lower grade. Over an hour and a half, 2-hour session, it dawned on both of us that if we were to bring these two elements together, we were actually sitting on a combined technology offering with some pretty radical implications across the board.
I think one of you told me at some point, I think Hong-Ming, maybe your process had kind of 10 unique steps. Originally, you were relying on very commonly used kind of bags and flasks, these kinds of technologies that are very used very widely across the world. When you map the IRO system or the IRO process with Farlan, that sort of 10 steps reduced to like 6 steps, including your novel vector. So tell me a little bit about how you were thinking about the efficiency gains. I know we'll talk about what actually happened when we started working together, but as you were thinking about it, what were you hoping to get out of the collaboration? I know certainly COGS reduction, human resource reduction was top of mind, I'm sure.
Yeah, so the goal is actually, I was thinking about it, is to try to make the product in hospital bedside, right? So, and for that, and again, we need really simplify. And first, we need to make the process really simple, like the really a nurse or a non-PhD technician can operate. So a complex CAR-T production typically involve early leukopak process, isolate T cell with beads, and then activate T cell with another kind of beads or antibody. And then after that, you have the debeading, right? And then you washing and add the virus, and then transduction, washing, and then that's actually initial full step, right? It's very complicated, and then there's a prone to error, and then transduction efficiency can be variable, right? So we think that step is a need to simplify, and we're thinking if we can eliminate that T cell isolation step, activation step, and then you can strictly act the virus to the leukopak, and then you can really simplify, and you can do it strictly in one bioreactor without change vessel in between, without washing. So that we think is going to be make the end game is possible, make a better side T cell manufacturing possible. We're getting much closer if we able to do that.
Absolutely. And you touched on a couple key points there. So part of our underlying thesis at Ori is to de-skill the labor that's required and fully reduce. So we reduce on the average process, we reduce by at least 60% the amount of labor required. But importantly, the other 40% is de-skilled. To your point, you can have a technician level, kind of high school graduate level person rather than a PhD immunologist that's in hard supply, or a nurse could use the system who's got many other duties and many other responsibilities. So this idea of de-skilling the process, but also being able to use it outside the clean room potentially, because IRO is a closed system. We wouldn't have to have a high grade space in the hospital to manufacture, and that's a critical element to potentially broadening out access and trying to operate in a decentralized model, which could have benefits, particularly in a large country like China or the U.S., where patients have to travel long geographies to get to treatment. So we wanted to test some of this thinking as we move forward with this partnership. If I recall correctly, we installed the system at your lab, so one in the PD lab, one in the GMP lab, around December 28th. I think it was almost the end of the year last year, if I can
Yes,
recall.
it's Christmas time. Yeah.
That's right. That's right. Some of our team spent Christmas in Shanghai with you guys, but we trained your team on the ground there in a week or so. And then the work started, the sort of collaboration of trying to map your existing process onto the IRO system, and then begin sort of optimizing from there. So maybe, Farlan, you could just walk through that process a little bit. And Hong-Ming, I'd love to get your perspective on kind of how that went. And, it took place in a relatively short amount of time, I think. So Farlan, if you don't mind.
Everything moved incredibly quickly. We managed to get an instrument out to Shanghai, 2 instruments actually. We did our IQ-OQ installation in Hong-Ming's lab. We set up one of the instruments in their PD lab and one of the instruments in their GMP lab. Team at ImmuXell were trained very quickly how to use the IRO instrument. I think it took about a day or two. And Hong-Ming's team were up and running the process that they wanted to take into the clinic basically a couple of days after the instrument was installed. So that's the key first thing. How quickly can you go from installation? Every moment is absolutely critical. We're in a rush here right across the board. How quickly can you go from installing an instrument to running a process in a GMP setting? That happened incredibly quickly, and we were all sitting together on Teams, both in Shanghai and in London, looking at the biological output as the run progressed, looking at the cell numbers, looking at the oxygen profiles, looking at the transduction efficiencies. So in a very short amount of time, we were getting a good feel of how IRO was performing with Hong-Ming's process.
I mean, Hong-Ming, you and I were discussing this not that long ago. Tech transfer usually takes 9 to 12 months, maybe, if you're moving quickly. Transferring your underlying manufacturing technology is often a complicated process. When you first saw the collaboration between our team and your team, and how quickly we were able to get up and running and start to get some biological results, was that surprising for you? I mean, I don't know what your expectations were, but I think it would be a surprise compared to what people normally would expect.
I think I'm really personally surprised it's so smooth, so fast, because I used to be CSO in another CAR T company. We try to move our production process from one side to another side. In the same city, it takes us 1 year, right? So it's very difficult, even within the same company, two different manufacturing sites. So here we have something, right? Move from another country, not only totally new, right? For us, is we never do a closed automate system before. And then we actually added up complexity using a new virus, right? We are not using our old generation. So it's a two different challenge. And then that we're able to do it in 4 months is amazing, actually. I think it speaks for the robustness of the machine, right? Already made. And it's really it's not only flexible, right? So it can be adapted to different kind of protocol and different kind of virus. It's also very robust because it can show in any kind of cell. So we're also doing TCR-T, which require usually 1 or 2 log more cell than typical CAR T. So again, right, we're able to generate enough cell for patient after I think 451, right? We're testing different condition and able to generate cell enough for TCR-T for solid tumor, and that speak for how reliable and still flexible of the system are we building.
Yeah, you threw a bunch of stuff at us, right? So
Yeah.
we had a new virus. We hadn't done a huge amount of work with retro. Farlan, we've done
Yeah.
a little bit, but not a lot. We had a TCR, which requires a lot of cells and a long
Yeah.
process. And so, Farlan, I think one of the things maybe the audience wouldn't appreciate yet is the kind of collaboration that your team would have with a partner like ImmuXell. And as you said, we were monitoring the run data remotely because it's a connected system from London, talking with Hong-Ming's team every day about what was happening, offering ideas. And, we also did some, I think, work with our computational fluid dynamics model along the way to see if we could optimize mixing. Maybe just describe how that collaboration works in practice. Would be useful.
The process that Hong-Ming just described is not a simple one. We have a novel virus that's never been used before. And on top of that, the virus is responsible for not only the transduction, but also for the activation and the selection. This is where IRO really flexed its muscles. It was able to develop a process which was very gentle at the front end, required some very complex biology at the front ends to do all of those different processes simultaneously on day 0. But at the same time, as you heard Hong-Ming mention, with these kind of therapies, you need to get to a lot of cells. So to go from a very gentle early stage process to a process where, in an ideal world, you'd have a stirred tank bioreactor or something like that to grow up enough cells to reach dose. And that's really where our system was able to help, is to do all of those things in one single container. To get something complex like that working quickly, it was tremendously important to have our team in London and the team in Shanghai working together. In London, we have a very experienced, highly skilled fluid dynamics team. So at the heart of this is, how do you mix the cells with the different viruses? How do you get the cells out of the lag phase and get them growing? And then how do you reach those very high cell numbers all in the same systems? You have different mixing regimes to do all of those different things. So our team in London were working side by side with the team in Shanghai, and we're actually doing biological runs in London. So, in London, we also have a very skilled T-cell biology team. That close working together, aided, of course, by the fact that IRO is cloud enabled. So as we're doing our runs, we're getting real-time information about what's going on in the bioreactors. Both teams were reacting, from one day to the next as we knew how each run was going. So yeah, it's not just the technology, but it was the coming together of the two teams. I have to mention the scientists on both sides are very talented. The leads in Hong-Ming's team and the leads in Ori's team were really able to accelerate together. And then they had the technology, both in the virus and the manufacturing system, which enabled them to move at that pace. So I think that's a kind of a summary of how things went and the kind of underlying reasons for us being able to move with such a complicated process so quickly through PD to a place where we could start thinking about putting it into the clinic.
So the collaboration continued through the first 3 or 4 months of this year, 2026, from its kind of origins at the end of December 2025. Maybe there were 4 or 5 test runs or optimization runs, PD runs, to really understand how the two systems, the kind of innovation from ImmuXell combined with IRO, could be optimized. And then I think it was sometime in April, Hong-Ming, you and your team felt comfortable starting the process to manufacture for the first patient.
Yes.
So tell me a little bit about that. Kind of, your program and the IIT program is targeting solid tumors, so specifically pancreatic cancer, but also looking potentially in the future or maybe currently at colorectal cancer. How did you think about, you're working with the investigators across China, and so talk me through the kind of patient selection process you go through, and then what actually happened in the manufacturing process. I know you ran your standard process and the IRO process in parallel. Talk to me a little bit about that kind of that work that you undertook.
First of all, we actually work with the top hospital in China, and both major city, and they are really top expert in terms of we call digestion system tumor, right, including pancreatic, colon, and liver. And in China, there's many patients, and then typically for pancreatic cancer, there's uh, no good option, right? So when they develop metastasis, and there's a great need, so we are easy to get doctor excited when we see cell therapy can work for the pancreatic cancer. So we set up a system in one of hospital. They really have more than 1,000 patients, right? And they see every year, and then we decide work with them. And then one of the benefit of IIT in China, I think, is more flexibility. So usually it's quite difficult to if you already set up a production procedure, right? A protocol, not easy to change or improve it, right? So in China, we still able to uh, building flexibility in IIT. So when we find something we believe can work better, more efficient, we can do the comparable study, right, in a lab, and we show it's comparable in terms of what do we believe is the quality of the cell, and also need the safety standard we routinely do. So we're able to make 2 batch typically for each patient. And then if they are comparable, so we can infuse the cell we think is will perform better for patient. So I think FDA also thinking that way. Not extensive study needed for phase 1 trial, and the process can be flexible as long as they're compatible. So we have the advantage of that. So really speed up, right? So we can not only we generate data quickly, but also able to make have all PI right now how to set to using the new cell producing in using Ori machine, and then again say, right, so for the first patient, we see a very similar expansion profile. It's safe, patient don't have any problem or no extra side effect, and then we typically see advantage of TCR-T, right, in terms of safety. So it's relatively safe compared to CAR-T, and then we're able to convince the PI to do it. And then from now on, we know it's safe, so hopefully we can follow up that with some more patient in next few months.
The process, as I understand it, normally, so when we manufactured the dose for the patient, that was in early May, late April, early May, 2026, I think. Two of the things that we most often are working towards with our partners are higher transduction efficiency and faster cell growth. Obviously, to get to those higher cell yields, or particularly transduced cell yields, we need both of those things. So your normal process, as I understand it, it's a couple weeks. Is it about 12 to 14 days, something like that?
Yes.
What did you observe when you ran the two parallel processes? One in the kind of your legacy Flask-based process, and one in the IRO system. What was observed by the team as you went through the manufacturing?
Our data is still early, right? So we actually compare both BAC and then T-Rex, and it's always zero about reactive. So from our first few batch, we feel at least they're comparable. So as long as we have the right condition and cell expand perfectly, right, in three systems. And all these systems, as Jason say, it faster. So it does faster. But we still try to optimize. Farlan already told us, right, there's a many way we can improve the transduction. And the reason we need to careful study because the virus is totally new. So require a different kind of mixing during transduction. And that we still try to improve. And we think our efficiency can even better. But so far, it's comparable to our standard protocol. And then so works both a little faster than our typical bag or T-Rex system.
Excellent. And that leads me to kind of the next question. Just wanted to speak a little bit about the future of the collaboration. So where do we go from here? I know we've identified another patient or two that we hope to treat in the IIT, and you spoke, Hong-Ming, just then about sort of additional optimization we want to do. Maybe Farlan, if you want to just lead on kind of the next steps of the collaboration with Hong-Ming's team from the Ori side.
I think the most exciting thing about this collaboration for me is understanding how process improvements impact not just on cell growth and transduction efficiency, but also on measures of efficacy. I think one of the things that's happening in this collaboration is we will be doing some in vivo tests to have a look at what's the killing capacity of the cells that we manufacture in different manufacturing conditions. But also, we're going to have the clinical data coming from each patient. So as we iterate and as we improve the manufacturing process, we're going to be getting that all-important in vitro and in vivo functionality data. And I think that's critically important in these collaborations. In this field, efficacy hasn't always followed logic, what you think would happen logically. I think it's very important to have that data feeding into your understanding of your manufacturing process during PD. So the IIT system for me is it's the ideal platform for developing cell and gene therapies because you have that all important functionality information to inform which direction you want to take the process.
And Hong-Ming, from your perspective, what's where do you want the collaboration to go? I know you've got an ambition to file an IND later this year and to treat many more patients. So tell us a little bit about that and how Ori can support you.
Finally, make a very strong point about uh, what we can see, right, in terms of the cell, and how can we correlate to uh, efficacy. So far, from what I understand from all the CAR-T or TCR therapy, uh, we still don't know the critical quality attribute of the cell. For example, we really don't know the dose, right, and the phenotype, and all these kind of questions. We still don't have clear answer. So the collaboration between all the immune cell, we might able to do something like that. So for example, our old bag or G-Rex, we don't collecting much data. So with always bioreactor, we have real-time monitoring and all collecting all the data. And one day, maybe we combine with machine learning AI model, we can determine the critical quality attribute, right, of the cell, and then we can actually guide us how we improve our process. And that's one collaboration I will be very excited to work and also very important. Another I want to see is can we build a platform, integrated platform, right, with our virus and bioreactor and possible the robotic arm, right? So able to build a platform really to see whether we can do it in a decentralized system and in country like China, with much reduced cost to deliver what we our cutting edge cell therapy to patient in much wide population, not just a few rich people. So that's two front I want is a scientific front is to know what's kind of cell we need to generate, and then and second is try to deliver right to more patient at a reduced cost.
Absolutely. And Farlan, this is one of your major platforms that you like to talk about is sort of the ability to modularize and scale up, and then add robotic automation on top to really get to those tens of thousands of doses. I mean, I think pancreatic cancer has 40,000 patients a year, I believe, diagnosed. That may be a U.S. number. I don't know if that's a global number, but it's a very large number compared to what we are producing today as an industry. I think last year in 2025, the whole industry produced 12,000 doses, at least of the approved products in the U.S. So, how does that vision that Hong-Ming just articulated, Farlan, or fit into Ori's ambition to scale up production in the future?
Me and Hong-Ming are totally aligned on scale up. The ability to add a layer of robotics onto multiplexed racks of Oris really opens up the ability to generate a very large amount of doses in a small amount of space. And I think what's really exciting in China at the moment is the way in which cell and gene therapy manufacturing facilities are being embedded from the very start into new hospitals. I think that is really, for me, going to drive access across China and will serve as a model for the rest of the world. Ori's ability to work in PD, as we talked about earlier, but also to be multiplexed depending depending on the room that you're in. Are you in a room in a hospital? Are you in a large? Do you have a large facility for centralized manufacturing where you can apply robotics? All of those different scenarios are well suited to the Ori platform, and I can see them being adopted in all three different modes in China. This is the kind of thing we want to do. We want to do process development and therapy development using tools like AI. We also want to scale up manufacturing so that the therapies can be delivered in clean rooms in hospitals across China. But we also want a system where if we want to centralize manufacturing and use robotics to really hit those high numbers, we're going to be able to do that as well. And it's so exciting to have a therapy developer working hand in hand with advanced automation to deliver what I think this field needs, which is combined discovery and manufacturing platforms. When you bring those two things together, you have the ability to develop the blockbusters of the future.
One of the takeaways from our visit recently to visit Hong-Ming and his team in Shanghai and around China, when we visited that brand new hospital, it was 3,000 bed hospital that had an embedded 800 square meter cell therapy manufacturing facility inside. It was purpose built to deliver essentially localized manufacturing. And I think the fact that access is constrained in China because of costs, as it is similarly everywhere. I think the numbers that I saw of the approved products that are sort of the Western products that have come to China, maybe 2,000 patients in total have been treated over the last 5 to 7 years that they've been available. But there's 2,500 different IITs happening around China, and hospitals like the one we visited and innovative companies like ImmuXell are driving that innovation, because ultimately patients need access to these therapies, and the market will find a way such that we can ensure that we can deliver safe and effective medicines for these patients. So I'm proud to be aligned with a partner who shares our vision of enabling widespread patient access to these life-saving cell therapies, and impressed with the incredible momentum, Hong-Ming, that you and your team have at ImmuXell and the innovation that you're bringing to what are very, very difficult diseases, as we know, as you said earlier: pancreatic cancer, colorectal cancer, liver cancer. These diseases don't have very good treatments today, and patients have unfortunately, a grim prognosis often when they're diagnosed. So this offers hope to those patients and their families. And so, thank you for doing that, and thank you for your partnership. One last question I had for you is: what does the future hold for ImmuXell? I know you've got a potentially exciting other partnership in the future, and you might be taking on a bigger role. Can you tell us a little bit about that?
Yeah, so for ImmuXell's, so it's a company of 5 years. So we, uh, I think we're very proud we're able to do right so far, and then we are lucky to have a collaboration like Ori, and then lucky to now somebody can help us to put us on the international stage. So we're going to file a new IND, hopefully, very soon in United States, or with collaboration with the UK, maybe in Europe too. So our company looking to into get into we call overseas market, and so very happy have somebody able to supporting us. And one of the goal certainly to pushing our pipeline, right? And the second, probably really what I are very super lucky to work with Ori is to try to build the platform. Really, that's my dream too, to see whether I can build a system, integrated system, as we already talking about, to combine our discovery, right, our biology platform with the engineering platform. And then typically, I think nowadays with all the AI back system, and only definitely pioneer in that area to make us understand deeper, right, the biology and the cell we're making. And then looking forward to next couple of years, we will see, right, our product getting the patient, and then looking to build a platform not only can be used by us, right, and are able to offer to other uh, uh, cell therapy provider, right? To see whether we call a platform can allow more patient get a cutting edge product. That's our goal for near future.
It's a great point to end on. Thank you, Hong-Ming, so much for that and for your partnership. And we look forward to our work together in the future.
Thank you, Jason. It's a great time to have you come to China again, right?
I'll see you in a few months. We'll be there. So I'm looking forward to that.